Welcome to Maximizing Molecular Biological Analysis. Today we're going to take a look at the different ways that you can improve your molecular biological analysis. We all know that DNA and RNA are very important to the scientific community. Whether you're doing crime scene analysis and forensics, or you're working with microbiologicals like viruses and bacteria, or if you're doing food and taste testing or consumer safety testing and you need to test for possible food pathogens or other types of microbiological pathogens. The one thing they all have in common is that all these different types of samples generally have a very similar RNA or DNA workflow. A molecular biological workflow sort of looks like this. You select your target. What is your pathogen? Which sequence of DNA are you going to work with? You choose the type of sample. What is the form of the sample? Is it tissue? Is it an agricultural product? Is it saliva, blood, urine, or something like that? Then you do some sort of lysis of the cells to extract the genetic material. You isolate your genetic material using reagents, buffers, and DNA or RNA kits. You prepare it for your processing with primers, plates, buffers, and things like that. And then you do your replication or you do your measurement using PCR or qPCR. And that's going to be the focus of a lot of what we talk about is the endpoint of PCR and qPCR. The first step in the process is always sample homogenization. So if you have a heterogeneous sample, maybe you have different types of tissue, maybe you have tissue from different parts of an animal or different parts of the body or different parts of the plant, and you want to be able to test fully that organism, then you're going to take representative samples and then you are going to homogenize that into one sample. An important first step in homogenizing your samples is really understanding the physical parameters of your sample. What is its toughness or hardness? If you have a very tough sample, it's going to take more force in order to break it apart for particle reduction and ultimately homogenization. What's its structure? Is it abrasive? Is it a material that's going to start breaking down your equipment? Do you need to change the focus of how you do your particle reduction? Is it sticky, meaning that the traditional method you were planning maybe is not the right one because you're going to have samples sticking everywhere? What's its density? What's its moisture content? Some particle reduction or some homogenization pieces of equipment use fine screens. Well, if you use a screen with a wet material, you're going to clog the material. Conversely, if you have a dry material, you want to be thinking about, can it become airborne? Is it going to float in the air? Is it going to float within the vessel that you put it in and not become crushed? What's its melting or softening temperature? Is it going to degrade if you have to use liquid nitrogen? Or is it going to degrade if the temperature is too high? Is it going to clog? Again, are you going to clog those screens? Are you going to clog the mechanism for grinding? And finally, what purity of material are you concerned with? What is your contamination level that is acceptable? If you are using the material in a way that you have no real error or no real room for any contamination, that is also going to affect the strategy of how you decide to grind and homogenize your samples. Those physical forces are really going to determine what type of particle reduction you are going to need. There are four forces: compression force, which is two opposing forces coming down vertically on a material, and then you have them crushed in between those two opposing forces. You have a shearing force, like a scissor, where it actually just kind of cuts. This is things with blades. You have attrition force. You have horizontal plates, the materials in between them, and they are crushed between the movement of the plates together. So that's kind of like a grinder. And then you have impact force, which is when the material is hit by a force like a hammer, so like a hammer force or a ball for a ball mill. So when you have these four forces, there is some overlap, but generally, if you have very large particles that you need, you want things like crushers and cutters. Smaller particles, you're looking for attrition and impact mills, ball mills, and things like that. It also depends on those physical parameters. If they're very hard, you're probably going to be using a crusher. But if they're very soft, you're going to be turning more to the grinders in the mills. Again, if they're wet or malleable, again, you're going to be using grinders and mills more than you're going to be using crushers that have very structured materials that they're employing. Another thing you have to think about is if you're working in the lab, think about some very common tools. How about a mortar and pestle? Well, that's sort of a cross between a crusher and an attrition, but you have to know that you're going to be basically introducing contamination into it, and you're not going to get a very uniform particle size per se. So that is a mortar and pestle, so it kind of fits among the middle of the compression and the attrition. What about a laboratory blender? That's more of shearing force, so it's kind of more at the top near the cutters. So that's where that's going to be. And you're also going to have to worry about contamination. And if you're worried about metals at all, you're going to have to worry about metals contamination from blades. For the most part, analytical samples kind of live in the world of ball mills and impact mills, that small particle reduction. Well, some people say, do I even have to homogenize? And if I do have to homogenize, do I really need to use an automated system? Well, there are very good reasons for homogenization. One of them is the amount of sample that you need for homogeneity. If you have a sample that's about 5 millimeters, and that's the size of basically a pencil eraser, and you have a 10% uncertainty allowed in your process, then you're going to need 125 grams of sample. But if you bring that down to the 0.5 millimeter range, which is the size of fine point pens, then you really only need that 0.13 grams of sample. If you want a smaller uncertainty, like 1%, then you need a lot more sample. So we like to say that the sample preparation is really the first most important step to get homogeneity, to get a good extraction, to get efficiency. You preserve your compounds, you stop them from being subjected to any heat, and you reduce your chemical and your genetic changes, and they're very high throughput, and it reduces contamination. When you look at automated processes for molecular biology, you'll see that an automated process gives you complete grinding and very fast processing, opposed to manual processes, which in our experience can sometimes leave incomplete grinding. You can see the pellet that's still there in our sample on the right, and they can take hours to days to process, where if you automate the system properly, you can do many steps in your automation, so you reduce the time that you spend processing samples. This is a study that was done for DNA isolation using manual processes and our Spex genogrinder. As you can see, the total time for the sample process was 3 hours and 24 minutes for the manual homogenization, where for the genogrinder, it took us 28 minutes. And when we looked at the DNA concentration by UV vis at different types of tissue, we had mouse heart, chicken breast, and beef loin. In each case, if you automated the process, you put it in something like our genogrinder, our ball mill, you were able to really increase your DNA concentration, and you got better quality DNA. You got much cleaner DNA. You got much more DNA when we were doing our different gel techniques. In doing homogenization, I want you to keep in mind some sample processing considerations. They are basically temp, time, particle size, and pH. And for today's discussion, we're mostly going to be talking about temp, time, and particle size. In regards to temperature, there really is a sweet spot for molecular biology. It tends to be in that 10 to 40 degrees C range. If you start to go higher, you start to incur some cell death, and eventually you'll denature the proteins. If you go lower, again, you start to initiate cell death, and you get cold enough or low enough, and you'll start to rupture or freeze the cells. Often this depends on time. How quickly it reaches that high temperature, how quickly it reaches that low temperature, or how long it will be kept at that particular temperature. Depending on the type of analysis you're doing, there is a range of temperatures that they can be exposed to. So if you want living plant and animal cells, cells that will continue to live, then you want to keep them near that sweet spot. And there are some cases where you can take a little higher or a little lower, but definitely within that sweet spot. Living molds, bacteria, and viruses, again, It mostly hovers around the sweet spot with a little bit of variation higher or lower depending on the type of mold, bacteria, or virus. We've all heard the stories of the different types of bacteria and viruses that are found at extreme temperatures in the Arctic or at the bottom of volcanoes. So we know that there is a lot of variation, but for most pathogens, things that we are concerned about as human beings, we're mostly looking in and around that sweet spot. If you only care about DNA and RNA, you have a lot more flexibility. You just have to worry about the point where it's going to denature or the base pairs are going to separate. And that's where you are concerned with the higher temperatures. But we also know that you can flash freeze like with liquid nitrogen and that you are able to still recover usable DNA and RNA. It's also kind of similar with chemicals. If you're doing a chemical analysis, you are mostly concerned about not getting the chemical too hot. You don't want it to volatilize. You don't want it to change its structure, but usually colder temperatures are good. And of course, when we use liquid nitrogen, it's all the way down at the bottom of our temperature scale, some of the coldest material. But usually that's a very quick process when you do liquid nitrogen. It's almost an instantaneous freezing process. So that actually cuts down some of the damage across the board because it's an instantaneous freezing. Speaking of liquid nitrogen, if you're using it, you're probably using a cryogenic mill of some kind. And this will give you very fine analytical powders. It takes the tissue or the material that you're trying to grind and it embrittles it using liquid nitrogen. So if you're doing DNA and RNA analysis, it's very, very good for that. If you're doing chemical analysis, it's also very good for that. If you're looking for live cultures or culturable material, or if you're looking for live bacterium or live molds, then it depends on the particular mold, bacteria, or virus that you're trying to test for. And if you're looking for live plant or animal tissues or cells, then that could be more problematic because you can lyse cells or break open the cells and you will not have live material. Another approach is to use a laboratory mill or a ball grinder or an impact mill like we have at Spex. Now these are very good, but you have to worry about temperature because just by their very nature, any type of grinding process creates energy and energy creates heat. So any of these methods where you do not have some temperature control or you do not consider your temperature control, then you can actually have problems, especially if you have to grind it for a fairly long period of time. So the longer you grind something, the temperature can become a real issue when you're grinding your samples because grinding creates heat and friction. So you can quickly generate enough heat to suddenly cook your sample. And especially if you have to have a fairly long grind time. Maybe you're doing a particularly difficult type of tissue, maybe a fibrous piece of tissue, and you need to grind it for a significant period of time. Well, we did a little study. We have the ability with our Genolite to have cooling. So we took 3 scenarios. Uncontrolled, so no cooling whatsoever during the grinding. We cooled it to 5 degrees C, and we also cooled it to -5 degrees C. And we took plain water, and we ran the plain water over 2 minutes. And during the course of the 2 minutes, we took temperatures at different points. What we found is that when we cooled our system, we were able to consistently keep our temperatures cool. So we were able to keep our samples from getting above 10 or 15 degrees C. So we were definitely within that nice safe zone. The uncontrolled sample, however, very quickly rose in temperature. So we went within a minute to almost 40 degrees C and within 2 minutes to 40 degrees C. And now if we would have left this longer, it's quite possible this would have climbed into some dangerous temperature areas, especially for different types of cells. Now, so 40 degrees C, that was just for water. But if you imagine you have a more fibrous tissue, or you have a tissue that's going to generate a little bit more energy as friction is involved, you are very quickly going to start to have to worry about your temperature. So before we said that really, if you're going to use cryogenic temperatures, you're good for chemicals and DNA, but then you have to be concerned about your living cells and your living organisms. But if you're talking about a ball mill or other laboratory type of grinding equipment, it really is going to worry about temperature. Now, if you're able to cool your sample, either have a system that has a cooling as a feature,
Or sometimes you can pre-cool your samples. Maybe not put them in the freezer, although in some cases you can. You want to cool the samples down before you start to grind them. Put them in the refrigerator, use a little dry ice around them, and then you're able to keep them on the cooler side. You'll get more time, and then you will have more success over all the different types of organisms and tissues. However, if you are not going to employ some sort of cooling, then you are going to have to worry about how hot your samples are getting and how hot those tissues and those organisms are getting, especially if you need live organisms. Particle size is often something we really don't think about. If we're doing chemical analysis or we're doing molecular biological analysis, we usually just care that it's a very fine powder. Well, for Spex, very fine powder can be anywhere from 10,000 nanometers all the way down to the lowest report I was able to find of 10 nanometers. That's a pretty big range. And it's really a big range if you are looking for live cells. If you are trying to keep your cells alive, then you really need to know about particle size and how much you're grinding those tissues down to or those samples down to, because it's going to matter. If you are going to try to keep white blood cells alive, and white blood cells on average are 25,000 nanometers, and you grind your sample down or you process your sample down to a fine particle size of about 5,000 nanometers, you're not going to have live cells. You are going to have cell parts because you're doing cell lysis. If you are concerned about mold spores and you're grinding your particle size down to 1,000 nanometers, well then, of course, you're lysing your mold spores, you're going to be spilling cell contents out. You're not really going to have a lot of live mold spores. You're going to have mostly parts from the cell lysis. So this becomes very important if you're trying to keep cells alive. If you're doing molecular biological techniques where you're only measuring the DNA and the RNA like PCR, then you really don't have to worry so much about the particle size because you're not necessarily looking for live cells, but you are looking for the snippets or pieces of DNA. And then as long as you keep your particle size above 2 nanometers, which is the width of DNA, then you are able to keep those little pieces of information, those little pieces of DNA and RNA intact, so you should be able to use it. Now, most laboratory mills, as I said, are under 100 millimeters down to the micron level, but they're even down, as we can see, to the nanometer level. And what you have to worry about is the longer you grind a particle or homogenize a particle of, or your samples, that's where you're going to get smaller and smaller particles. So if you're homogenizing a sample for 1 minute, you might be getting 10,000 nanometer particle size. If you go down and you do it for another minute, you might be getting 5,000. If you might do for a couple more minutes, you might be getting all the way down to that 100 or below nanometer size. So time becomes part of the big issue. So when you're going through the different sizes, you want to make sure that you program and create a program that gives you exactly the particle size with the timing that you need in order to get the particle size that you're aiming for. After you perform your homogenization and now you have your sample, you're going to now process it for your DNA or your RNA analysis. You're first going to collect your samples, your tubes, your vials, your grinding media, your homogenization. Now, I have grinding media because sometimes you will use that homogenization equipment for your sample processing as well. So not only for homogenization, you use it for mixing as well. You have your chemicals, which are your lysis buffers, your solvents and your salts, and then your materials for the molecular biology. In this case, I'm giving the example of PCR. So you're going to have your primers, your polymerases, your nucleotides, your enrichment solutions. So these are all the things that you're going to have when you're doing your molecular biological processing. For something like purifying DNA, you are first going to do some digestion, and in this case, something like Pro-K digestion. Then you are going to have to separate out your DNA from the rest of the cell material, and in many cases that means binding it to some type of column. Then you're going to pass the lysate through and you're going to wash your sample, and then you're going to lute off your clean DNA. You can do this individually, or you can buy kits like our Spex DNAmax kit, which has all of the components in and the spin columns so that you're able to quickly purify your DNA. We did a little comparison of our DNA kit against some of our competitors, and we used our automated method with our geno grinder. We had some liver, some spleen, some chicken, and some beef loin. And you can see that our Spex geno grinders, along with our Spex DNA kit, our DNAmax kit, performed extremely well across the board. We were very comparable with the top selling kits, and in fact very favorable against some of the kits. So we had very good recovery across all the different types of tissue. When we looked at the gels for some of these extractions, we were able to find that we had very good extraction for a wide variety of materials, both plant and animal. And this is some examples of some plant material for cannabis and for pine needles. You can see that our return or our extraction recovery was very high for both types, especially compared to our competitor. And we had a very good quality of DNA from the gel. The nice thing is that we also get good DNA from this. So if you do spectrophotometry, you want to see a nice sharp peak at 260 nanometers. And you can see that our kit really has a nice sharp peak. You don't want to see is a broad band across many different nanometer wavelengths, or a very thin band at the wrong wavelength. That you can see on the right there is a wavelength at 230. So this is not good DNA. If you look at our DNA from our DNAmax kits, for our cannabis and our conifer needles, nice peak, nicely shaped peak, and right on that 260 and 270 wavelength. So you've got a really nice, good response for DNA. We believe our Spex family of homogenizers and our DNAmax kit are the perfect fit together. They're the perfect combination of DNA or RNA extraction, time savings, money savings, and good, efficient results. We like to say that the DNAmax will give you less time at the bench, so you have more time to spend on other activities. You get more samples in less steps with lower cost and a higher yield. You're not wasting all your time and your energy doing your processing by automating your processing and using our DNAmax kits. We're going to take a look now at what you can do with your isolated DNA and RNA. Modern scientists have a lot of choices when it comes to molecular biological analysis techniques. And some of them have been around for a very long time, like our cellular techniques, our cultures. Now, they're very sensitive and they're very reliable, but they tend to have low specificity. They're pretty time consuming, very laborious, kind of expensive. And if you don't have those living cells, so if you've processed your sample to such a small particle size or in a way that has killed off your cells because of temperature or friction or particle size, then you're going to have some false negative results because you're not going to have enough living cells to create your cultures. Then you have your immunological techniques. They pick up on the antigens, the ELISA methods. Again, very, very easy, high throughput, but again, some low specificity and low levels of detection problems. Finally, you have your nucleic acid, the ones based on the RNA and the DNA, like your PCR and your qPCR. This you need a very small amount of sample, and you don't necessarily need live organisms to do your PCR. So there are some costs involved, enrichment, purification costs, but you are able to work with samples that do not have living cells. So you are able to get your results quantitatively from samples which have been processed that do not longer have living cells. The basis for PCR, of course, is DNA or RNA. DNA and RNA, we think of them as the backbone of life, of biological organisms, but there's also a chemistry to it as well. The backbone of the DNA structure is the phosphate group paired with the deoxyribose sugar. So these two molecules fit together in a way that naturally, as they join one after another, as they connect one after another, they start to form a slant or a curve. This stacking and curve causes the twist that we know of in DNA. So that's that twisting of the DNA, that double helix kind of configuration. The actual genetic information is part of the master code from 4 chemicals, 2 purines and 2 pyrimidines. For your purines, you have adenine and guanine, and your pyrimidines, you have thymine and cytosine. And they pair together because the bonds align in a certain way that make them partners. So the adenine, the purine, combines with the thymine, which is the pyrimidine, and those bonds, those 2 points of bonding, come together over and over again in the DNA code. For the other pair, you have the cytosine, which is the pyrimidine, and the guanine, which is the purine, and they bond at 3 points. So they come together in 3 points and they make that bonding. So it makes them a lock and key as well. So this combination of these 4 base pairs, or for these 4 chemicals that make up the base pairs, create the master code for DNA. For RNA, it's slightly different. You only have 1 strand for RNA. RNA is often used as a carrier for genetic information. So within the cell, there are different types of RNA, and there is messenger and transfer. So there's different types of RNA, and they each are involved with transporting or creating the copying of DNA. So they are very important molecules, but they are a single strand. And instead of deoxyribose sugar, they have a ribose sugar, so it's a phosphate and a ribose. Also, what you find is that thymine is replaced by uracil. So you have adenine then paired with uracil, and the cytosine again is paired with the guanine. So you still have that pyrimidine-purine pairing, but you have a different pyrimidine in uracil. The cells act like factories to copy the DNA. In a cell, the process of transcription and translation then produces multiple copies of DNA, or genetic material. The DNA double strand unwinds, it denatures. The area that is going to be copied is then transcribed by messenger RNA. Then it is brought to the ribosome in the cell, where the transfer RNA and the proteins then make a copy, sort of like a typewriter, adding little pieces along the way, and you get then the copy of the mirror or the reverse of the piece that was transcribed. So then you have a copy of the original portion of the DNA. So this is sort of like a little factory, and when we're working with PCR, something very similar happens. PCR artificially does the same type of process that occurs in the cell when DNA is transcribed. So you have your DNA strand, and by denaturing it between 94 and 98 degrees, you separate it into 2 separate strands. Primers are then chosen to start the process of the copying or the translation of the DNA, but they are specific to whatever target. So they're basically the marker for the start point of where to start and you stop your copying. So the primer then is very specific to the selection of the area of the DNA that needs to be, or the RNA needs to be copied. So the primer then attaches, then you anneal your sample between 50 and 68 degrees C. This is kind of temperature sensitive depending on your primers and your polymerases that you're using. And then the polymerases come in, and then the primers are added during elongation, where the primers then attach to the subsequent base pairs that are being formed, and they make those base pairs as they go along. So they reconstitute the base pairs with the nucleotides. And what you end up with are 2 copies of the DNA strand at the area which you are trying to replicate and that you're trying to amplify. That process is performed over and over again in order to amplify the DNA into the millions and millions of copies. So the first time you do it, you have 2 copies, then you have 4 copies, and then exponentially you keep going up in the number of copies. So the first step we said is the denature. You open up that DNA strand and that 90-something degree temperature, the DNA strands separate. Then you attach your primers at the annealing portion, and then you add your nucleotides as you extend, and you build that new strand. And then you repeat over and over again: denature, anneal, extend, denature, anneal, extend. And it's all controlled by changes in temperature. just a minute ago, every time you perform this cycle, you double the amount of DNA that is produced. So from that first strand, you get 2. From that second strand, you get 4, and so on and so on and so on. So you keep doubling the amount of DNA. There does come, however, a time where you basically plateau out. And that's usually limited by the amount of chemicals, primers, nucleotides that are provided for the reaction. So you get your lag phase, that's when you get that little small amount of growth, then you hit that start of exponential growth in your log phase, and then finally, as you start to run out of materials, you hit that plateau phase. And that will take any number of cycles, anywhere from 20, 30, 40 cycles or so. So it will keep on continuing until you basically run out of material. There are 2 types of PCR. You have your endpoint PCR. This is your conventional PCR. This is where the only purpose for the PCR unit is to double the DNA during every cycle. So you keep doubling it over and over. It heats, it cools, it heats again. It heats, it cools, it heats again. It doesn't have any way of quantifying the DNA. You usually use another process like gel electrophoresis. So at the end of your thermocycling, you use gel electrophoresis in order to determine the DNA sequences and the quantity that you've amplified. In real-time PCR, or qPCR, it's also called quantitative PCR, it's a little more advanced. It can actually count or quantify the DNA as it's being produced. So it has a fluorescent marker that's employed. It's usually within the nucleotide master mix. And this fluorescent marker, along with a series of sensors and lights, can then measure as the DNA is being produced. So then a measurement is taken and you can see the amount that's being measured every time a cycle completes. So whenever it is amplified, you'll get a hit with the fluorescent marker, and you can see this over and over again as the amount of DNA increases. There are many applications for molecular biology. Some of the most important ones are testing for pathogens, things like E. coli and Aspergillus and penicillin. So these become very important in industries like food and agriculture and pharmaceuticals. So we're going to take a look at the application for the testing of pathogens. As human beings, we are exposed to potential pathogens all day long. Think about everything that you take in your life, that you're exposed to in your life. The food you eat, the pharmaceuticals that you take, the water you drink, what you breathe, if you vape, what your environmental exposures are. So these become the pathways for which we must monitor our different pathogen levels. There are many agents for disease for foodborne and waterborne illnesses. In fact, there are over 250 specific agents, and they can range from industrial pollutants to poor sanitation to poor hand washing to food handling. So you first have your physical contamination. This is your large debris, your dirt, your bugs. Then you have chemical contamination, where maybe pesticides, heavy metals, or other materials seep into food and water. Then you have parasites. These are worms and nematodes and other types of organisms that enter the food or the water and infect human beings. And then you have your microbiological contamination, your bacteria, your viruses, and your molds. Now, if we look at worldwide foodborne illnesses and deaths, and this is according to the World Health Organization, there are over 300 million foodborne illnesses a year. And that means that 1 out of 10 people in the world will experience some sort of foodborne illness. But it also means that 420,000 deaths a year occur worldwide due to foodborne illnesses. In the United States, there are 48 million illnesses a year due to foodborne illness, according to the CDC. That means 1 in every 6 people suffer some form of a foodborne illness. And that equates to 3,000 deaths a year due to foodborne illness. So what are the targets when we're looking for our microbiological pathogens? Mostly we're talking about bacteria, viruses, and fungus. And of course, there are also other organisms, but these are the three primary ones that cause a lot of illness. Around the world, there are quite a few pathogens of concern when it comes to foodborne illness. Viruses like norovirus, bacterium like Salmonella or Campylobacter, viruses like hepatitis A. And then there's aflatoxins, which are actually a chemical agent produced by fungus. So the highest death rate around the world, 89.4%, were from aflatoxins. But there were no human cases here in the United States, and so there were no deaths. But botulism, which is a bacterium, had the highest death rate in the US of 17.3%. So these are definitely pathogens of concern when it comes to foodborne and waterborne illness. Earlier we talked about particle size, and we said that the particle size was going to affect some organisms depending on their size. Here's a relative chart of all the sizes of the different pathogens of concern around the world. You have hepatitis A, which is very, very small at 30 nanometers. Then you go all the way down through your Campylobacter and your Salmonella. Your Salmonella is 5,000 by 1,500 nanometers, and your botulism bacterium is 8,000 by 800 nanometers. So these are fairly large size bacteria. Now, remember the chart we looked at about particle size. If you are grinding your sample down to kind of like in that talcum powder to that clay size, so something in between in that 10,000 to 5,000 nanometer range, then you are going to possibly destroy some of these agents. So if you are going to grind it down to the smallest size over a long period of time, you might be possibly damaging or destroying the smaller agents. If you're grinding to a medium fine particle to very fine particle size, then there's a possibility you might be destroying or killing your Shigella, your E. coli, or your Aspergillus. If you're going even larger particle sizes, anywhere from 5,000 to 100,000, then you might be killing off some of your Salmonella or your botulism. And it all depends obviously on time, how long you grind it. The longer you grind your sample, the smaller the particle size and the bigger the chance you're going to lyse these different cells and you're going to end up with non-viable or non-living cell cultures. Earlier we talked about temperature. Each of the different pathogen agents has a deactivation temperature and time, how long it's exposed to that temperature in order for you to kill it or for it to be deactivated. For Shigella, if you're testing it, it's 55 degrees for 60 minutes. Well, imagine our little experiment where in 2 minutes with just water, we got up to 40 degrees C. How quickly can we reach 55? Is it going to be a minute? Is it going to be 2 minutes? Depending on the tissue, it could very easily climb to that 55 degrees in a very short amount of time. Now, is it going to stay at 60 degrees for 60 or more minutes? Probably not. But you are going to start damaging the cells, and you are going to kill off a certain portion of the cells, which is artificially going to lower your culture results. Now, if you're doing PCR, you're not as much affected by that. It's not going to really matter if your organisms are killed or if your sample really gets a little warm. So this is not going to be as big of a problem for PCR, but if you're using typical culture techniques, you are going to want to be concerned on what temperatures are you generating and how close to the max temperature do you have to get before your sample becomes non-viable for the culture you're going to do. Some of the most common mycotoxin species of fungus are Aspergillus, Penicillium, Fusarium, and Claviceps. Now, these are all very famous species of mycotoxin producing fungi, and I'm going to show you a few classical examples in the next slide. But primarily, for when we're talking about PCR, I'm mostly going to be talking about the Aspergillus and in the future, the Penicillium species. So we've all heard of some of these cases of mycotoxin poisoning. One of the most famous ones, we've heard of Curse of the Pharaohs. So supposedly there is a possibility that when Howard Carter opened up the tomb of King Tut, that they were exposed to these mycotoxins. There were these black molds. So that was one of the possible mycotoxin poisonings. The Salem witch trials. We've heard about the Salem witch trials, and during either the 60s or 70s, some research was produced that showed that maybe this was due to ergot poisoning, which is by the claviceps species of fungus. So it's possible that it caused hallucinations when this particular fungus grew on the rye and the different grains that they would harvest and eat and make into bread. In Kenya in 2004, there was an outbreak of aflatoxin and ochratoxin poisonings, so mycotoxin poisonings. And then we've had many, many dog food and pet food recalls over the years due to mycotoxin toxicity. And of course, as I said before, black mold, you've all heard about black mold. Any homeowner who's gone to buy a house has had some concerns about, is there mold in the house? Is there black mold in the house? Because these all produce potentially poisonous mycotoxins. Two of the most important groups of mycotoxins are the aflatoxins and the ochratoxins, and those are primarily from the Aspergillus and the Penicillium species. So with these two, they produce some very deadly toxins, and these are concerned in a large variety of different agricultural products and cannabis products and things like that. So these are of the most concern and often the most regulated. Now, the good thing about these aflatoxins, these ochratoxins, is that the toxins themselves are pretty hardy, even if the mold spore is not. But if you're not doing PCR, then you do have to worry about being able to detect the aflatoxins, the ochratoxin species, because if you kill them off, you're not going to get a good culture. Now, of course, if you are using PCR, then you don't have to worry if your Aspergillus species actually survived your sample processing, or if it was exposed to too much heat, or you ground your particle size to too small, because they ultimately use fragments of the DNA, and you are able to detect the Aspergillus using PCR. So you're able to quantify your Aspergillus and get an accurate count on your Aspergillus in your agricultural sample. Spex is also working on additions to our Pathogen Kit catalog. This will include things for the food and beverage industry, like Salmonella, or E. coli STEC, and total yeast and mold, all intended for use with the PCR. If we put together all that we've looked at in this presentation, it's pretty straightforward. First, you want to automate as many parts of your process as possible. That includes mechanical homogenization, the use of kits for DNA and RNA extraction, and if you're doing pathogen testing, the use of pathogen kits. All of this helps automate your process, it speeds it up, and makes your time more efficient. Then you want to optimize. You want to optimize those processes so that you get the right homogenization method that is for your sample target and the structure of your sample. And you also want to be cognizant of the temperature, the time, and the particle size, because as we saw, those can actually have an effect depending on what your final process is going to be. If you're going to be doing PCR or plating or ELISA, you're going to want to know what the temperature, the time, and the particle size are, and if those are going to affect your final outcome. All of this creates a maximized process in which for you to do your molecular biological work. Thank you.